Good morning. This is the screencast that corresponds to the lecture of Monday, May 14. I'd like to finish with some concepts that we talked about in the molecular biology class. So this is the general schematic of the molecular clock of mammals. And one thing that I said is that one way in which this, this is how the clock indeed remains as an autonomous oscillation, right? But if the clock only did that and didn't control the transcription of other genes, this should not be of much use. One way in which the clock can regulate the transcription of other genes is because BMAL and clock, the dimer, can bind other E-boxes in other genes, and that way turn transcription on. And because the activity of this dimer is rhythmic, then all the genes that will have an E-box in principle should be regulated in a circadian fashion. We also said that the clock has the ability to shape chromatin remodeling, so that also allows for giving access to DNA or not to RNA polymerases to undergo transcription. So there are many different mechanisms by which the clock can control the transcription of other genes. And in the same way that we call all these genes that are involved in this loop clock genes, and that is, by definition, they are genes whose transcription and translation is essential for the normal working of a circadian clock, and they're part of this loop of the circadian transcriptional, translational feedback loop. So this is a transcription, translation, loop, or TTL, as you saw it referred to in the paper by Tomita et al. In the same way, as there are clock genes, there's also what we call clock control genes. That is, these are genes whose transcription and translation is also rhythmic, but they are not an essential part of the molecular clock itself. In other words, if you knock out one of these genes, or in principle if you knock them out completely, all of them, the clock should still be running. However, if you knock out one of these clock genes, the clock stops. As we said, so that's in general how transcription and translation is controlled outside of the loop. Another thing that to put in the broader context is that although this is the main way in which most circadian clocks are made with a TTL, there are other mechanisms to generate circadian rhythmicity, and you've already seen one in the Tomita paper, in which they demonstrate that there are circadian clocks that do not rely on transcription and translation, but only on cytoplasmatic changes in post-translational modification of proteins like phosphorylation. So that's a non-TTL clock. Okay, it only relies on cytoplasmatic events. It doesn't involve transcription or translation, but that's in principle should be considered as an exception. We only know of two cases. One is cyanobacteria, and that's a paper that you read. The other one is in red blood cells, which have no nucleus, and interestingly, they can also have circadian rhythms, so those circadian rhythms cannot be explained by transcription, and in principle neither by translation, because these cells don't have a transcriptional machinery. So I said that a great percent of genes in your genome, in mammalian and all the animals that have been studied, genomes are indeed under circadian control. And in fact, I show you this slide that shows the genes that are transcribed in a rhythmic fashion. So again, just to give you a sense of how to read these plots, these are genes stacked up vertically. So each one of these little rows, and there are many rows, is a different gene or a different transcript in principle, and how it is expressed throughout the 24-hour cycle under either a light-dark cycle or under constant conditions. And what you can see is that all of these genes have a rhythmic expression. So red would be high expression, green would be low expression, and high again, because there are 2 days of recordings. It's not a double plot, but actually real 48-hour recordings. And what you see is that whereas some genes will peak in the middle of the day in expression, some other genes will peak in the middle of the night. So there's a different phase of expression for each one of the genes. I also said that the set of genes that cycle in the SEN, for instance, is different from the set of genes that cycle in the liver, and only a small % of them are common to both tissues. And in fact, altogether, we said that up to 70% of the genome may be under circadian control if you take all the tissues from all your body. So this just, again, another example comparing liver and skeletal muscle. We also said that if you knock out clock genes, like in this case, it's not a knockout, but the clock mutation. Remember that the clock knockout is rhythmic. What you see is that this very nice rhythmicity that you had, I think in this case it's in the liver, it's mainly lost when you just mutate the clock gene. So that means that if you somehow disrupt or break one of the components of the molecular clock, of course, not only you impair the clock work, but you also impair the transcriptional regulation of all the clock control genes. So these are all clock control genes, or CCGs. Okay, then we also went through the molecular basis of familial advanced sleep phase syndrome, and we said that it was a single point mutation that led to a change in an amino acid, which is the amino acid phosphorylated by casein kinase 1 epsilon, which was the protein coded by the tau gene. Okay, so by changing that amino acid, casein kinase epsilon cannot phosphorylate properly that the period 2. This is a period 2 mutation, okay, and therefore the period of the period of animal changes in the same way that the period of the tau mutant change, but for a different reason. In the tau mutant, the mutation is in the casein kinase epsilon. In the human disease, the mutation is in the PER gene, in the period 2 gene, where the casein kinase acts. We also said that, in fact, you can, and well, this was a question, remember why you should be able to tell that this is a problem in period and not in amplitude or light input. If the person doesn't have light input, it will be free running. It won't show an advanced phase of entrainment, which is what these patients have. But if the person has a different period, what happens is that if you have, so a normal person that has a period longer than 24 hours, right, would, much longer than 24 hours, would need to make the light coincide in the advanced portion typically. So that means that they need to expose their phase response curve to light late on the subjective night, and that will lead to advances. But if your period is closer to 24 hours, then that means that you will need to expose a different part of your phase response curve, right? And in this case, what happens is that these people have periods that are shorter than 24 hours, and they need to expose actually their phase delay portion. Therefore, they need to get the evening light to hit deeper in their early subjective night. This is early subjective night, late subjective night, right? So, which means that in order to do that, they need to, in a way, push their night. So their night is here, their subjective night. They need to push it forward a little bit so it gets exposed to the evening light, which means that they will go to bed earlier, right? And they will wake up earlier, because everything, the sleep bout will move together with the phase response curve, okay? So essentially now light, instead of being here for these patients or here, it will be more like there, right? And everything will be occurring a little bit earlier for these people. So that's in a way how understanding the phase response curve tells you that small changes in your circadian period, particularly if they switch from a period that is larger than 24 to a period that is shorter than 24, can have dramatic changes on your phase of entrainment, and this is why these people are going to bed 4 hours earlier than normal normal people without the mutation. And this is proof of principle that happens. The main two panels that you need to concentrate on are these two. This is when you express the same human mutation in mice in a background of per 0, you get a very extreme short period. If you express it in a background of wild type per 2, so this is per 2, 0, 0, right? I mean, minus, minus, plus the human FASP gene, per 2 gene, and in this case it's a per plus the human FASP gene. And in both cases you get a shortening of the period. It's more dramatic when you don't have the wild type copy of the per 2 gene. This is just adding the human gene to the mouse. But the point here is that if you express the human gene, the human version, the mutated version from these families, you also shorten the period in mice. So you get the exact same effect that you're getting humans. Okay, so now we move to a new and fascinating topic of circadian biology, which is how we synchronize to the light-dark cycle. That is how the circadian system gets information about light, or circadian photoreception. So, as I told you, this is the basic schematics that we started the class with, where we know that because of the Earth rotation, there's a input pathways that communicate the information about the LD cycle, the light-dark cycle, to the clock itself. We know the clock in mammals is the SCN, and there are output pathways that produce, for instance, rhythms of sleep or activity that are shown here. And those rhythms, although they are driven by this endogenous clock, they are entrained to the light-dark cycle. So this is dark, this is light, and this is a nocturnal animal. And you can see that this is true entrainment, because when you release the animal into constant conditions, there's the phase is the same as the previous phase. So how does this happen? Well, it took quite a while to figure out, at least in mammals, how this happened. I told you that there was this very interesting case of a blind person in which essentially this blind person, as opposed to other blind people, had normal circadian synchronization. And this is data from your paper, in which they show you the overall pattern of activity for about 2 months, a little bit more than 2 months, for 2 blind patients. But in one case, you see that the entrainment of one of the patients is not as good as on the other. And the way you know this is because every once in a while, you take them into the lab and you take their melatonin minimums or temperature minimums, and you can tell what the real phase of these patients are in their circadian system. And you can see that, in fact, their clock is free running, because these melatonin maximums or temperature minimums are free running. Whereas in this patient here, they are entrained to the 24-hour light-dark cycle, right? So obviously these 2 patients in their normal life, remember, you're bringing them to the lab only every couple of weeks, but the rest of the time they're undergoing their normal living. So this clearly indicates that one patient is entrained to the light-dark, or to something, maybe not to the light-dark cycle, but definitely entraining, and the other patient is not. And in fact, as you see in the paper, we know that these patients indeed can also inhibit melatonin, and this was a big surprise and could not be explained. By the end of the lecture, and by the time you read this Hatter paper for Friday, for this past Friday, given that this screencast is for May 14, you will see what the mechanism is for synchronization in mammals. There are a few possibilities. One possibility, of course, is that these patients are using known visual cues to entrain, and actually cues. In fact, some blind patients do use social cues, maybe food, entrainment, and other inputs to synchronize. But actually, most patients that doesn't quite work very well. The other possibility is that there are visual cues, and of course, or photoreceptive cues, we should say. And this should be non-photoreceptive or non-photic. Okay. And if they do use photoreceptive or photic cues, right, in principle, those could be going through the eyes or through other centers, right? And, of course, for us, for humans, it's very hard to conceive that we can perceive light through organs that are not the eyes. However, you will see that in other vertebrates, in fact, this is the case, and it's not unthinkable of. And in fact, in the Zeisler paper with these blind patients, they had to prove that if they blindfolded the patients that inhibited melatonin and that entrained these blind patients, they could not respond to light with inhibition of melatonin, demonstrated that indeed although they were blind, the information about light was going through the eyes. Okay, so let's see a little bit of history of examples that indeed demonstrate that light doesn't necessarily need to go through the eyes. This very old paper from 1961 that was working with aphids. Aphids are these insects that suck up the sugars from plants, and basically they can be entrained even if you shine light outside of the eyes. And the conclusion of this paper was, one might in light of such evidence be cautious about assuming that photoperiodic control is mediated by obvious photoreceptor structures in any given case. So basically they realized that in principle the eyes of these organisms were not necessary for entrainment. And people said, well, that's interesting, but these are still insects. And in fact, people wanted to explore whether other insects would have the same property. And using Drosophila as a model, in this case what we have are flies that have the per luciferase contract, per look, in their whole body. And what these authors did is to isolate parts of the fly. You can dismember this poor fly in many ways, and then you can either have the head alone, as shown here, okay, or you can have an antenna, as shown here, a wing, and then you can put them on a dish, keep them alive, and measure per luminescence, per driven luminescence rhythm. And you can see that they can show beautiful rhythms. Not only this demonstrates that they have a peripheral clock, which by now should not be a big surprise for you because even we have peripheral clocks. However, the very striking part of this is that they can be entrained by the light-dark cycle, which is shown below, independently. In other words, you can shine light-dark cycles to the wing, to the antenna, to the head, and each one of them will be entrained to those signals. So that means that not only these parts of the body have the clock, but they also have the input, the molecular input necessary to synchronize that clock. Okay? And now we actually, and knowing what you know about the Drosophila circadian clock, and using, happens. In fact, I told you that in Drosophila, one component, not of the clock, but one component of the input to the clock was cryptochrome. Cryptochromes spell cryptochrome. Cryptochromes in flies and plants are photoreceptors, and they mediate input to many different systems, but in Drosophila, cryptochromes are important for the synchronization of circadian rhythms. And in fact, the way this works is that cryptochrome itself is in the same cells where the molecular clock is. So in order to reset the clock, all you need to do is shine light directly to the clock cells itself, which is different to what happens in the suprachiasmatic nucleus, for instance, because the suprachiasmatic nucleus doesn't see the light. And we need to transduce that with glutamate, as we saw previously. But in here, what happens is whenever light arrives to this clock cell, it will stimulate cryptochrome, and after stimulation, cryptochrome will bind to TIM, and by binding to TIM, it will induce its turnover or degradation. So basically, eventually, TIM will be gone. When PER is hanging out alone in the cytoplasm, it also is gone. So basically, this brings the PER TIM levels down. Okay, so if this is PER protein and TIM protein levels, whenever you stimulate with light, because of cryptochrome that binds to TIM, then the levels will go down to 0. So this is interesting in itself. But however, how does this lead to a real phase shift? Well, it turns out that the per TIM dimer is up at night. So if you have the light-dark cycle or the subjective day and subjective night, and you measure per TIM levels, they look like this. This is per protein and TIM protein, okay? So they are peaking during the nighttime, which means that if you bring them down in the early subjective night, you will then bring back to the daytime, the late afternoon values, so that will be a delay. Whereas if you bring them down during the late subjective night, you will be bringing them down to the early morning values, which is essentially an advance. So you can see how, in this case, the grading per and TIM translates into phase delays in the beginning of the night and phase advances at the end of the subjective night. So this is quite different to what we saw for period in mammals, that we said that period was low during the night and would bring it up with light. So in this case, in mammals, you're bringing up a messenger RNA period that is low during the night. You're bringing it up with light, whereas in Drosophila, you're bringing down a protein complex that is high during the night. You're bringing down it in the middle of the night. But the resulting effect on phase resetting or phase shift in the clock is the same. That is that if you present light in the early subjective night, you will delay the clock. If you present light during the late subjective night, you will advance the clock. Okay, so that's how molecularly. Let's look a little bit of the neural pathways that are involved in photic entrainment. Well, here we have the first kind of surprise in invertebrates. In this case, these are blinded sparrows. And what you see here is that these animals can perfectly entrain even when they have no eyes. Not only they can entrain, but they can also show responses to long or short photo period. So sparrows are diurnal, obviously. If you put them in a 12-12 light-dark cycle, they show this short bout of activity, right, that matches the 12-hour light phase. If you put them in a long summer-like light-dark cycle, 18-6, you see that their activity matches that. They expand their activity pattern. They can also undergo phase delays, and this indeed represents real entrainment because they free run from that phase. So there's obviously no doubt that sparrows without eyes can actually entrain to the circadian system, entrain to the light-dark cycle, sorry. So how do they do it? Several experiments were done in sparrows to demonstrate that they have multiple photoreceptors in their brain. For instance, if you remove the eyes, right, but you have an intact pineal, turns out the pineal in the sparrow is not only a clock but also has photoreceptors itself. You get entrainment and normal photoperiodic measurement. Okay. So if you look at the reproductive axis of these sparrows, they will respond normally to photoperiod changes. If you remove the eyes and the pineal, both of them, then the animals cannot entrain, but they still remain intact in terms of their photoperiodic measurements. So somehow they can still tell the length of the photoperiod and the difference between summer and winter. Interestingly, if you have eyes but no pineal, entrainment and photoperiodic measurement is preserved, meaning that both the eyes and the pineal can have input to entrainment, okay, because in here, if you remove the pineal and the eyes, you had no entrainment, right? But then, so if you remove both eyes and pineals, entrainment is gone. However, if you leave either one of them, entrainment remains. And what's really interesting here is that if you take these animals that have no eyes and no pineal, and you cover the skull with ink so that light cannot get through the skull bone, these animals also lose photoperiodic measurement, which means that taking all this evidence together, there are photoreceptors in the pineal, in the eye, and also deep brain photoreceptors. So sparrows are amazing, and this is true for many birds, but interestingly, the influence of each one of these photoreceptors differs from species to species. In some species, you have more predominance of the eyes. In others, you have more predominance of the pineal, and so on. Okay, what about mammals? Well, in mammals, similar experiments where you remove the eyes and then expose the animals to a light-dark cycle have clearly shown that mammals without eyes cannot show entrainment. So in other words, all our photoreception to the circadian system goes through the eyes. Now there are some findings that demonstrate that we also have photoreceptors in the skin that could synchronize local skin clocks, and actually all that evidence is from Ethan Berg, who works here at the University of Washington, working together with Russ Van Gelder in the Department of Ophthalmology. But in any case, in principle, most of our circadian behavior is always synchronized by the light-dark cycle, which is relayed to the circadian system by the eyes. So in other words, we don't have, as far as we know, deep brain photoreceptors, or our pineal is not sensitive to light. Okay, so we know that light inhibits melatonin, and actually, as you've seen in the paper by Teisler et al., this is true even in blind patients, which was a big shock. How does this work? Well, the answer to this question came from mouse models. So before we understand how the photoreception works, we need to have a little bit of of anatomy of the retina. The retina, as you know, is part of your brain, and it's our main photoreceptive system. And basically, there are 3 layers of cells in the retinas that are functionally divided. One is the photoreceptor layer, or rods and cones, and actually it used to be called the photoreceptor layer because before we discovered melanopsin, we used to think that this was the only photoreceptor layer of the mammalian retina. There's an intermediate layer that is essentially taking care of neural processing of information that comes to the photoreceptor layer. And then there's the ganglion cell layer, which is the retinal ganglion cells are the only ones that project deep into the brain and convey all the information from the retina to the brain. So the classic view of the retina until about 15, 20 years ago was that, in fact, the only part of the retina that was photoreceptive was the layer that contained rods and cones. Okay. However, there was, by the way, one big surprise is that when you looked at what was the peak sensitivity for melatonin inhibition, and you don't need to worry about how to construct these curves, but essentially, when we looked at the peak melatonin the wavelength that was more effective in inhibiting melatonin at night, the color that was more sensitive was in the range of the blue, and it didn't fit any of our known photoreceptors. So in this case, what we show is what is the spectral sensitivity for each of the known photoreceptors in rods and cones. And basically what happened to here is that melatonin peak of sensitivity was kind of in the middle of two photoreceptors, but it didn't really fit any of the known photoreceptors that we knew back in the late 90s. So people were kind of puzzled by this, because usually if you look at some photic response, you should be able to explain it by the photoreceptors that are present in the retina. In this case, that was not the case. So what people decided to do is to start studying mouse models in which you could remove rods and cones, right? And that way, essentially, you render these mice blind. And as far as we knew, this should lead to mice that could not only not form images, but also have no entrainment and no other what we call non-image forming functions of photoreception. In other words, you can use photoreception to form an image, like read a letter, or seeing a tree, seeing colors, seeing contrasts. But you can also use light for non-image forming functions like circadian entrainment or the constriction of your pupil, in which in order to constrict your pupil or to entrain your circadian rhythms, you're not consciously perceiving light or shape. So in principle, these animals that had no rods and cones should have completely impaired circadian entrainment and melatonin inhibition, but that was actually not the case. So this is confirmation that the strategy worked. So this is histology for the retinas of wild-type animals and the retinas of these animals in which you induce genetically the degeneration of rods, and that also leads to eventually to the degeneration of cones. And you can see here that you have these beautiful stained rods and cones in brown because you use an antibody that labels both rods and cones. Then in this layer, you only label one type of cones, and in this other layer, another type of cone. But the point is that you see in panels A, B, and C that these retinas have the normal layers of the retina. By the way, this is by cutting. If this is your retina with the optic nerve, what you're seeing here is a cut through the retina so that you can see all the layers in a transversal cut. However, when you look at these mutants and you do the very same dissection and staining, you see that they have all the layers of the retina except the rod and cone layer. So essentially, by inducing that mutation, you wiped out all the rods and all the cones of these animals. And in fact, they confirm this molecularly by looking at first using southern blots, then using northern blots, and then using reverse transcription PCR to demonstrate that genes that are typical of, in this case, green cones or UV cones, or in C rod opsins, genes that are typical of rods, are present in the wild type in one mutant where you knock out cones only, but in the mutants where you knock out rods and cones, there's no expression of these genes. Okay, so this is just basically showing that you not only effectively remove the cells, but also these are highly sensitive techniques like PCR that tell you, is there any expression of the genes that are specific to rods and cones, And the answer is no, okay, with different methods. So you're 100% sure that these animals are not showing any rot and cone. They don't have any rods and cones. There's some maybe leftover expression here for the UV cone, but it's not a big deal because in fact for all the experiments, they make sure they don't have any UV light in their experiments. But in principle, you can trust that this is a very effective way to remove both rods and cones. And then essentially they did the experiment. They said, can these animals entrain or not? And if they put them in a regular light-dark cycle, they indeed entrain, but they even did light pulses in the advanced portion of the phase response curve, and this is the response that they get. So this is the wild-type animal, the animal that has no cones and also eventually no rods. And in this panel, they are also showing you 3 genotypes: the wild-type animals, the animals that have no rods and not cones, and the animals that only have no rods, but eventually as they develop, they also lose their cones. And basically what you see is that the phase shift that you get in response to a light pulse, this is the same way that you generate a phase response curve, but they are only showing you one time of the day, at one circadian time at which they presented a light pulse. And basically you can see that for these animals, the response of the wild types is undistinguishable from the mutant. They also did a group without eyes, enucleated, just to demonstrate that indeed, if you do remove the eyes, there's no photoreception. This is not significantly different from 0, okay? So essentially the animals without eyes do not have any circadian photoreception, indicating that whatever is giving the mutants information about light is in the eyes, okay? Even when they have no rods and cones. And then in this case, this is the same. There's no difference between wild types and the double knockout. And not only that, the double knockout at higher light intensities. This is, by the way, each one of these is one light intensity, and you can see that the phase shift is bigger as you go up. But that's both true for the mutants and for the wild types. The same is true here. However, at the very higher intensities, what you see is that the animals that have no rods and cones even have a higher response. This is different from this than the animals that are wild types, indicating that maybe rods and cones at higher intensities could have some inhibitory input, who knows, or maybe that there's a baseline inhibition that you remove when you remove the rods and cones. But the point here is that a retina that has, remembered, of the 3 layers, so these retinas have no rods, no cones. The animals that have these retinas can entrain perfectly to a light-dark cycle. They can also have a normal pupillary reflex, and amazingly, they are blind. So in other words, if you do any tests for image-forming vision in these animals, they are blind. So in a way, these animals are very similar to the first patient that visited Chuck Zeisler's lab, who was blind, who reported to be blind but could still entrain. So this is starting to give a hint of what's going on with these patients. At the same time that these experiments were finishing, there was a paper by Iggy Provencio first at Uniform Services and then at University of Virginia, that showed that there was a new photopigment that he first found in frog skin, but then he found it in the eye, in the retina of many different animals, including humans. And the interesting thing is that this protein looked a lot like a photopigment, that is, a protein that could respond to light, like the opsins that are present in rods and cones, but it looked like more like an invertebrate photopigment. But in any case, it looked a lot like a photopigment, but instead of being expressed in the rods and cones, which is what you'd have expected if those were the only photoreceptive cells, right? It was expressed in the ganglion cells. But interestingly, it was not expressed in every single ganglion cell, but only in a tiny percent of them. So about 1 to 2% of all retinal ganglion cells in mice and rats had melanopsin, or were melanopsin positive. Okay, so it was a very small percent, and because animals that have no rods and cones still had the ganglion cells intact, people suspected that maybe even when they were a small percent, these 2% of cells could still confer information about the light-dark cycle to the circadian system, to the pupillary reflex system, and to the melatonin inhibition system that is also going through the suprachiasmatic nucleus. So the first experiment, what they did is to knock out the melanopsin gene, and this was kind of a disappointing outcome because, you know, people thought, well, if melanopsin is truly the photopigment that is mediating photic entrainment, then if we knock out the gene, the animals should not respond to a light pulse. However, they responded with a smaller phase shift, but they could still phase shift, and they could also entrain normally to a light-dark cycle. So if you put these animals without melanopsin in a light-dark cycle, they would show the normal nocturnal activity. This is not shown in this actogram. But also, if you give them a light pulse, they show a smaller shift, but still shifted. Okay. Also, they also show relatively normal Aschoff rule responses. So the lengthening of the period under constant conditions that you see in mice with brighter light intensities was still there, both for wild types and for the mutant. The mutant, it's called OPN4, and this is opsin number 4 essentially, which is the melanopsin gene. Okay? So far what we have is that although melanopsin is expressed in these 2% of the ganglion cells, when you knock out the gene, the animals can still entrain. They don't do it as well as an intact animal, so that was kind of disappointing. So the notion that emerged from these experiments is that, well, maybe melanopsin alone cannot do it. You know, if you remove melanopsin alone, cannot do it because these ganglion cells, even when now they have no melanopsin, they're still getting input from rods and cones. Because remember, in these mutants, we only removed melanopsin, but we didn't touch rods and cones. So this channel of communication should still be intact. Okay? So that was, at this point, the explanation of why these animals could still see this. So the next experiment, obviously, as you probably imagine, was to use double mutants and to both knock out melanopsin or knock out rods and cones, and this is one mutation that leads to the degeneration of both rods and cones, or do both. And in these animals, what you have is, if this was the original retina, right? So let's see, the wild type would look like this. Rods and cones are intact, the intermediate cells are intact, and then all their ganglion cells are intact, and some of them, I'm going to draw them in blue, these blue ones express melanopsin, right? It's 2%, I'm exaggerating the percent here, okay? And there's normal entrainment. In this case, what you have is rods and cones, again, all the layers are normal. However, you'd remove the melanopsin from the cells in this OPN OPN4 minus minus. So the ganglion cells are here, but they don't express any more melanopsin, and still these animals entrain normally. In this case, you remove rods and cones, so basically what you have is you only have the intermediary cells and the ganglion cells. And you already know from the previous slides that these animals also entrain normally. However, in this case, you remove rods and cones. Oh, and by the way, in this case, some of the cells have melanopsin, right? So you didn't touch the melanopsin. However, in the fourth case, you have the combination of the two mutations. You remove the rods and cones, and you also remove the melanopsin. And in this case, as you can see, these animals cannot entrain at all. So you have them through a light-dark cycle. They cannot show any entrainment. They are always free running, even if you put them into a light-dark cycle. So these mice behave as totally blind patients. And what I mean by totally blind patients are patients that have lost both their image-forming vision, which is most self-aware blind patients, right? All self-aware blind patients, but they have also lost their non-image forming vision, that is the photoreception that allows them to synchronize to their light-dark cycle, synchronize their circadian rhythms and sleep rhythms, or to constrict their pupil. By the way, these animals are also not able to constrict their pupil in response to light, okay. And they're also not able to inhibit melatonin. By light. So obviously, when you remove the melanopsin gene and rods and cones, you render these animals totally blind, indicating that indeed melanopsin, when rods and cones are gone, is essential for these non-image forming photoreception function. Okay, so then this is just a demonstration that melatonin inhibition doesn't work, but instead of measuring melatonin inhibition, they're measuring AANAT mRNA. So this AANAT is the RNA that makes an enzyme, AANAT, which is what the rate-limiting enzyme to make melatonin. So without that enzyme, you cannot have melatonin. And in fact, the way in which melatonin inhibition works in rodents is by inhibiting the synthesis of this RNA. So you stop this, and you stop the synthesis of melatonin. So in this case, they are looking at how much, and you see that these are negative numbers, inhibition of the levels of RNA for NAT was there when we give a light pulse. So essentially, if you give a wild-type animal a light pulse, you see good inhibition of the RNA, so you reduce the RNA a lot with a light pulse. If you do it in the animals without rods and cones, the same is true. If you do it in the animal that has no melanopsin, the same is true. However, if you do it in the double mutants, there's no inhibition of RNA, which is equivalent to say that there will be no inhibition of melatonin release. Okay, so this is another way of probing or of investigating the melatonin inhibition pathway. So essentially what we have so far is that animals without rods and cones and without melanopsin cannot entrain their circadian rhythms, cannot have image-forming vision, cannot inhibit their melatonin, and cannot show pupillary reflex. So they are indeed totally blind. and this is data from the paper that you've already read, so I'm going to go very briefly about this. So far we have demonstrated that melanopsin is sufficient to entrain circadian rhythms. So if you remove rods and cones and you leave melanopsin intact, this can lead to normal entrainment and pupillary constriction and inhibition of melatonin. What this paper showed, and this was actually done before, was that indeed melanopsin cells projected to the relevant areas that needed to be reached by these cells in order to achieve circadian entrainment and pupillary reflex and melatonin inhibition. And this is the Tau-LAC-C strategy that you read in the paper, showing that indeed melanopsin cells target the SCN in a very dense pattern. They also target the intrageniculate leaflet, which is another area that it's involved in circadian modulation. And they also target the olivary pretectal nucleus. Sorry, I think it's olivary with an eye, like an olive. So the olivary pretectal nucleus is responsible for pupillary reflex. So here you can see that melanopsin cells reach all the centers that are critical for circadian entrainment and melatonin inhibition, because we also said that in order to inhibit melatonin, you channel that information through the SCN. Of course, for circadian entrainment, these are the projections to the SCN also, and then to the olivary pretectal nucleus for pupillary reflex. And we still don't know really what the projections to the leaflet do, but we know that the intrageniculate leaflet also modulates circadian rhythms. So you are responsible for knowing the general strategy that we use in this paper to demonstrate that indeed melanopsin cells reach these centers in the brain. And the other evidence that so by the way, one thing to keep in mind is that if you were to look at all the ganglion cells that we have in our retina, which are, thousands, or at least, thousands, most of them will go to what we call the visual thalamus. Particularly in animals like primates, where we are highly visual. A lot of the projections, the vast majority of your retinal ganglion cell projections will go to the visual thalamus, which then will go to the visual, these neurons in the visual thalamus will project to the visual cortex. And that's this pathway which involves most of our retinal ganglion cell and a great deal of our brain, both in the thalamus and in the visual cortex, particularly in primates, is for image forming photoreception. And what I mean by image forming photoreception, I mean being able to see colors, being able to distinguish contrast, anything that you are aware of seeing. However, that 2% of the cells that have melanopsin, right, that, we could draw it somewhere here among the other ones. And I draw them blue because melanopsin is a blue light photoreceptor. They go to these other centers like the SCN, the OPN, olivary pretectal nucleus, the IGL, and some other areas of the brain that we're still kind of learning what they do. So this is a much smaller percentage. However, these cells that project to these areas are the ones that are responsible for non-image forming. Photoreception. And in fact, when we first talked about track tracing in the retina, that we said that this was in a way how the suprachiasmatic nucleus was discovered, was by injecting a track tracer on the retina itself. That track tracer will arrive to all these areas, right? But it will arrive to the SEN and these areas mainly through getting into melanopsin containing neurons, okay? So these are just some general questions that you need to consider for when you review this lecture. Of course, I will answer these questions later in review sections or if you post them on the discussion forum. So a person with a lesion of the retinal ganglion cells will have no circadian entrainment, and have no image forming vision, will have no pupillary reflex. All of the above are true. Okay, so just think about the answer to this question and then we can discuss it later in class or on the discussion forum. This is a typical exam question, midterm question. A person with a lesion that includes the totality of the visual thalamus will have no circadian entrainment, and have no image forming vision, will have no pupillary reflex, none of the above. A person with a lesion of the olivary polytectal nucleus will have no circadian entrainment, and have no image forming vision, will have no pupillary reflex, none of the above is true. Okay, just think about these possibilities and we can discuss them later. And this is very similar to what's shown in your Hatter paper that you read. Is essentially showing that indeed having melanopsin in the cells confers them the ability to respond to light intrinsically. So these cells are intrinsically photoreceptive. If you think about it, all ganglion cells can respond to light because they get input from rods and cones. However, melanopsin containing ganglion cells are the only ones that are intrinsically photoreceptive. What does that mean? And this is developed in the Hatter paper. What it means is that these cells can respond to light even in the absence of any synaptic input from any other cell in the retina. And all these recordings showing depolarization and action potentials are shown, in fact, in the absence of any synaptic input to the ganglion cells, as you've seen in the Hatter paper. And in fact, in one case, I think this is the E cell. Oh no, H. H is a cell that is completely isolated on a petri dish on its own, that it's melanopsin positive, and you can see that it responds to light even when it's alone. So this obviously cannot be explained by input from rods and cones. So the point is that having melanopsin confers the ability of these cells to respond to light in an intrinsic fashion. So people thought, well, if this is true, it should be possible to express melanopsin in cells that are not photoreceptive and induce them to become photoreceptive. And in this example, what the authors did is to express melanopsin in a cell culture line which are derived from kidney. And of course, kidneys are not photoreceptive, right? Our kidneys do not respond to light. But this cell line that is originally from kidneys can in fact show melanopsin. And what they're showing here is GFP staining. This is green fluorescence protein. And what that's showing is that your construct to target the melanopsin to these kidney cells worked fine. So this is just a marker, a reporter of your construct. The cells are green. That means that they were genetically transformed with the DNA construct that expresses melanopsin. And then in red, you stain them with an antibody against melanopsin. And you see that indeed they are expressing melanopsin in the membrane as they're supposed to do, because melanopsin actually is expressed on the membrane. And this is the converged image with the two colors. But the point is that if you look at cells that you transfected with melanopsin, amazingly enough, they can respond to light. Even when they're originally kidney cells, only by putting melanopsin in them is sufficient to make them photoreceptive. And these are control cells which were transfected with GFP, but not with the melanopsin as a control, to show that photoreception is not just induced by putting any piece of DNA or any coding protein, but just by putting melanopsin. Okay, so up to now we know that if you remove rods and cones, entrainment and phase shifting is normal. If you remove melanopsin but you leave rods and cones, entrainment and phase shifting is normal too. Right? That if you think about this, is that when you remove melanopsin, in this case, somehow rods and cones are communicating that light information still to the suprachiasmatic nucleus. Which means is are the cells that are communicating rod and cone input to the suprachiasmatic nucleus melanopsin containing, or are they ganglion cells that do not contain melanopsin? So in the parallel hypothesis, what we hypothesize is that rods and cones send the light information to the SCN via cells that do not contain melanopsin. In the serial hypothesis, what we say is that all the information from rods and cones reaches the SCN through cells, through retinal ganglion cells, there's no other way, but that do have melanopsin. So in order to test this, what the question experiment did is, instead of removing just the gene melanopsin, they actually induced the death of all the cells that contain melanopsin. And essentially the way they did this is to use a similar OPN4 tau-LAC-Z reporter, which is the one that you use for the Hatter paper. But they also had a construct that was the OPN4 promoter, that is the gene that codes for melanopsin, driving diphtheria toxin, which is a toxin that will induce cell death. So essentially any cell that expresses OPN4 in this animal will in a way commit suicide. It will induce its own death. And basically what you can see is that in the wild type you see all these dots in the retina that correspond to, this is on a flat mounted retina, corresponds to melanopsin positive neurons, whereas in the OPN4 DTA animal, it's a heterozygote, you see only one cell, maybe if anything, but basically no cells that are expressing melanopsin. And here is using your reporter gene to label melanopsin cells. If you have OPN tau-LAC-Z, you see that there's these blue dots which represent the melanopsin cell bodies and the axons. However, if you do the same technique in the animals that have diphtheria toxin driven by melanopsin, what you can see is that there's no melanopsin neurons at all. So again, very important in this case, we're not just removing the gene melanopsin from the genome, but we're actually inducing the death of all the cells that express melanopsin. Okay, and the question is, can these animals carrying retinas without melanopsin neurons and trained to the light dark cycle? And the answer is no. This is quite amazing because these animals, so again, wild types, these are the animals that have no melanopsin and no rods and cones that we've already talked about, and below are the animals that have no melanopsin cells. So in this case, we remove the cells themselves. We sacrifice those cells, and you can see that these animals are unable to synchronize to the light-dark cycle. They are free running all the time. So this is really amazing because if you think about these animals, what they are is, in our schematic, they have rods and cones, right? They have the intermediate cells. They have all their ganglion cells, right? But they don't have the cells that have melanopsin. So in other words, these blue cells are no longer there, right? So it's not that we remove the gene melanopsin from the cells, but we remove all the cells altogether. And these animals can perfectly form images. They have intact image forming vision. However, they cannot entrain their circadian rhythms, meaning that the real hypothesis is correct. That is that all the input to the SCN goes through melanopsin ganglion cells. This is better in your schematic, but essentially all the input to the suprachiasmatic nucleus from rods and cones is coming from retinal ganglion cells that contain melanopsin, and the ones that do not contain melanopsin are responsible for vision. So this is another question that I want you to think about. Which of the following animals will be completely blind? A mouse without the melanopsin gene, a mouse without rods and cones, a mouse without the melanopsin gene, not rods and not cones, none of the above. Again, completely blind means that it doesn't have any kind of photoreception, circadian, melatonin, pupillary reflex, or forming. Which of the following animals will show no circadian entrainment? A mouse without a melanopsin gene, a mouse without rods and cones, a mouse without the melanopsin containing retinal ganglion cells, all of the above. Again, think about these and we'll discuss them further. And of course, we go back to our clinical study. This lecture started with this patient that amazingly enough was blind but could inhibit melatonin and could have normal circadian entrainment. Now, after about 20 years of research in mouse models, we can understand why some blind patients can actually have intact circadian entrainment. So that's it for this lecture.